Well, we've made it to round 8. We've talked about genetic variation, we've talked about hexadecimal, we've talked about gene expression, we've talked about annotation, proteomics, metabolomics, even a little flow cytometry. And finally, we made it to biological pathways and networks. Why do we put this last? It maybe shouldn't be, but in some respects, this has been a somewhat neglected part of our field. And it's ironic because pathways and networks have largely been our key to distilling biology from data. Maybe you think of sequence reads coming off a sequencer as data, and then the annotation that you produce from them might constitute the information of a genome, for example. But if you want to be able to distill biology, knowledge from that information, I think it's quite valuable to be able to incorporate what is already known about all these different genes and their associations with each other, the reactions that their gene products may catalyze, for example, in order to distill biological knowledge from these parts lists that we've gotten so good at producing. So today, we're going to take that adventure into biological pathways and networks. And of course, I need to give a lot of credit to Bing Zhang, who taught me an awful lot about pathways and networks during the times that we had our labs in the same department. So I owe him a lot for putting this together. So frequently, when we talk about pathways and networks, we tend to talk about pathways, which is to say sort of canonical collections of genes that belong together. Maybe they're all in the same Krebs cycle, therefore they get stuck together. Maybe they're involved in a certain kind of fatty acid synthesis, so they all get lumped together. But I want you to think of this as kind of a fixed set of genes that reflect what we've developed over the years in knowledge about how these major pathways work within our cells. And frequently, pathways have been where one or many people actually lived for their entire careers, patiently hammering away one reaction after another to understand it. We call Krebs cycle Krebs for a reason. You know, this is a person who was very devoted to understanding central metabolism. So when we talk about gene sets, when we talk about a pathway analysis, we're usually talking about a whole categorization where a bunch of genes have been lumped together in one drawer, and we say, these are the genes for fatty acid synthesis, for example. So when we talk about this, we're generally speaking about pathways in which we have a sequential relationship of these. We may be speaking about an ontology, and we mentioned that's kind of a special vocabulary word earlier on. But we may also be thinking about things that have a more amorphous relationship, one that's less clear in its structure. So for that, we tend to talk about network modules, which is to say we have a set of genes that tend to be co-expressed under the same circumstances their expression rises or falls. So network modules are one of these sort of inferred ideas, and their membership may be less canonical, that we know this set and only this set of genes is involved in fatty acid synthesis, for example. So network modules tend to be inferred based on the sometimes clique relationships that we have among those different members. Now, we're going to sort of leave networks to the end of the lecture, but we are going to spend a little bit of time talking about enrichment analysis. And as I mentioned this morning, this is one of the key things that we need to be able to do, which is to say, we have this set of genes we've deemed special. Maybe they're special because they were found to be differential in a cohort versus cohort study. Maybe they're special because we found that all of them contain the same sequence motif. There are lots of reasons that we would call them, we would call this particular set of interesting genes. Just because I talk about them as always being differences in a cohort versus cohort study does not mean it's the only place we get such lists from. So when you have a list of genes and you want to know about the enrichment of pathways and networks within them, it's because we want to understand a biological function implicit within that grouping of differences. Okay, for that we'll talk about WebGestalt's overrepresentation analysis based on the hypergeoemtric distribution, and in the latter case we'll talk about Gene Set Enrichment Analysis, which is a very widespread method as well, very influential. So where do we get all this information about which genes go together, which proteins function together, which proteins impact which genes, for example? And there are all kinds of ways that these relationships can take place. When we think about one researcher or a team of researchers laboring away for decade after decade on the same class of problem, like say Krebs. We might think of this as sort of the classical biochemistry, and we've certainly inherited an awful lot from classical biochemistry and understanding what pathways exist, what reaction precedes each in possible ways to get to a product. Microarray correlation is also a very big deal. Now, this is a point of some confusion for folks, so I want to try to nail this down a little bit. Imagine that you have been working with a particular cell line for many, many years, and you've discovered that under different physiological conditions, this cell line behaves in different ways. Maybe if you give it a bit of a hypotonic stress, it behaves one way. If you hit it with a drug, it behaves a certain way. If you grow it to stationary phase, where it's hard to get the nutrition to keep dividing and dividing, it behaves in a certain way. So imagine all these different kinds of ways that you might torment this particular set of cells. At the end of that day, you're going to have a lot of different scenarios under which you've evaluated its gene expression. So if you've done this, you may find that particular genes always show up hand in hand with their best buddies on that list. If you're dealing with E. coli, maybe you've got 4,000 different genes that might be active. You find that under conditions of hypotonic stress, this gene comes up, this other gene comes up. You note under conditions where a phage is attacking the E. coli, this one goes up, the other one goes up. This co-occurrence, this co-expression, then becomes a basis for our understanding of the correlation of their behavior. So correlation in gene expression becomes a suggestion that because their expression correlates, they play part in the same kinds of cellular processes. So think of this as if your best buddy is a bank robber and you're always seen in proximity to the bank robber, sooner or later the police might come to you saying, "Hey, are you a bank robber too?" This is more or less how gene annotation can take place in the case of microarray correlation. We see that this gene is always showing up alongside this other one, or just after this other one. So correlations are one of the ways that we infer a relationship to a kind of process. Okay, chromatin immunoprecipitation or CHIP experiments are another way that we use high-throughput sequencing. I know we've talked about just whole genome shotgun sequencing and RNA-seq and so on. CHIP is yet another way that we can do this. So if you want to understand about the relationship of a particular a particular antigen and the DNA that it interacts with, chromatin immunoprecipitation or CHIP experiments are one way that you can do that. But if you're going to do stuff like that, you might also do protein co-immunoprecipitation. So if you are able to pull down a particular protein and you see that another one is pulled out of solution with it, it may be that these proteins interact. They have a specific interaction that they always, for example, function as a heterodimer, where the active protein has two different polypeptides that are interacting with each other. So these co-immunoprecipitation experiments in proteomics are one of the ways that we learn about associations between proteins. Now, just to cut a very long story short, I would point out that sometimes when you pull down a protein, you get another one, not because they have a specific interaction, but because one of those proteins is sticky. This stuff happens. So you could get a false association, a false positive association between two proteins simply because one of them just really likes whatever you're pulling things down with, among other things. Okay, so protein IP is one. Yeast-2 hybrid is a system that we've used a lot for determining whether gene products interact. And yet, yeast-2 hybrid has something of a high false positive rate associated with it. So we can say that two gene products interact because they are positive on yeast-2 hybrid, but I think most people in their minds would probably have a little star on that to say, oh, well, this was just known by yeast-2 hybrid. We're really not sure. We've not seen it in other studies, for example. And of course, co-localization. So it's one thing to say that these two genes interact because they come up and down in the same conditions. But we might also say that these two proteins interact because they're always found, for example, in cell division, appearing at the centrioles. For something like that, you might say to yourself, well, now they're co-located, so it seems quite likely that they're involved in the same process. Okay, so these are different modes by which we might say, these things, they belong together. So these all provide different relationships between pairs of genes, or genes and proteins, or proteins and proteins, for example. And from that, we can deduce lots of different possible architectures for them. So when we say canonical pathways, we're really talking about this diagram on the wall. We have lots and lots of very well-known pathways that lots of organisms make use of. For example, glycolysis. Lots of things like splitting 6 carbon sugars in order to produce 3 carbon sugars, thus deriving a little bit of energy. And glycolysis can be found in all kinds of different organisms. So these canonical pathways have come down principally through classical biochemistry, often with a little bit of augmentation from various methods in systems biology. We can also get point-to-point networks. So if you know, for example, that when you pull down protein A, you frequently see protein B coming along, you can infer that these two are interacting in some way. So you could, instead of saying, well, these both play a part in this larger process, thus putting them on a pathway, you could simply say, it's enough at this point to know that A interacts with B, and not care so much that B then interacts with C, with D, with E. So we can infer these larger networks by noticing the point-to-point relationships that we have, the protein-protein, or gene-gene, or gene-to-protein, or vice versa relationships that we might have. And of course, we have these well-settled ontologies. So an ontology, as I've mentioned, is the combination of a controlled vocabulary, we're going to look at some examples of what I mean by a controlled vocabulary, and a set of relationships. So the ontologies show up in an awful lot of different disciplines. And frequently, the studious bookish types in your fields are the ones that navigate to this. So people who are kind of your natural librarians, people who like to have their knowledge nice and orderly. That's kind of the natural living space of ontologies. So we talked a little bit about gene ontology this morning. We're going to come back to it later. So what are the kinds of things that spin out of this? Well, I've drawn out a bunch of these. We're going to talk at some length about overrepresentation analysis and gene set enrichment analysis. But we will also spend a little bit of time talking about cytoscape visualization, because this is one of the really major tools that people make use of in systems biology. Network module detection is something that we might use to try to discover new relationships among genes, to try to recognize that, for example, if genes A, B, C, and D all have relationships to all three of the other, each have a relationship to all three of the others, we might call that a clique and say, these 4 genes are very tightly connected, they form a network module. Similarly, you might use something called a random walk assessment. You might ask, basically, how many gene-gene or gene-protein or protein-protein interactions do you need to step through to get from this gene to that gene? And you might use the number of steps as a proxy for how closely related they are in a biological network. So lots of ways that these can play out, and I don't mean to leave you with the impression today that only overrepresentation analysis figures into understanding those. So let us move ahead. Let us start with the bald assertion, I think that has been pretty clear all along, that lots and lots of these omics studies, genomics, transcriptomics, proteomics, metabolomics, that these all can produce interesting lists. We've seen our volcano plots, I think everyone remembers those. So we see that if we have a gene up here in this red area, which is to say it has a nice low p-value and it has a nice large magnitude fold change, that might be an interesting gene, so we could make that part of our list. We might have looked at gene expression over a whole variety of conditions and then try to cluster these genes based on similar expression patterns. This is the correlation of expression factor that I was talking about. So from this, you might also end up with a list. Now over here at the right, I don't have a list of genes per se. I actually have a list of probe names. So these are probe names from a microarray. and those in turn act as a proxy for a particular gene name. Okay, so you might say to yourself, gosh, gene 1 has been seen to be part of this particular pathway, but somebody else has claimed it's also part of this other pathway. Gene 2, well, I've got, it has a similarity with gene 1 in that both of them have been called part of pathway 1, but it also has been invoked to be possibly part of pathway 3. Gene 3, well, it kind of splits the difference. It's been found to be part of pathway 2 and pathway 3, and finally gene 4, well, it's only found associated with pathway 3. So now, how do I figure out which pathways I actually trust from that mess? So one of the things that you can do is ask which genes support which pathways. So in this case, we're simply reorganizing the data. Instead of asking what pathways would each gene be associated with, we're asking which genes on our list are associated with each of those pathways. This gives us the ability to jump to information about which pathways are the biggest players a little more directly. So why would we go about doing this? I mean, it's kind of an error-prone process, right? We don't have annotation for each gene being associated with all pathways. We don't have perfect knowledge. Well, in fact, we can use what's known about pathways and networks, for that matter, to leverage existing biological information. The curators of databases like gene-gene interactions and protein-protein interactions, the people who are behind this, are really working hard to harvest information in the peer-reviewed literature and bullet that down as a reference in this database that this pathway, that this functionality is associated with this gene. Why not use that? This represents institutional wisdom. If we assume that we're going to get all the information we want from each experiment, we're really cutting off our arm and leaving it behind. There's an awful lot of research out there that's already been encoded in these databases for us to take advantage of. So if we want to interpret our data well, I would say make use of institutional wisdom. You can interpret your data better if you are able to translate lists of interesting genes to interesting biological themes instead. So translating your result into a meaningful Go term is one of these things that people do in order to make their data more relatable to other people. Certainly, improved robustness is a huge advantage. So imagine that because of random variation, you were able to detect that these 3 genes were differential in your experiment, but you missed seeing another couple. We know that can happen just by the very large numbers of genes we look at. So you might have missed seeing a few genes that are differential just by random chance. But if you use pathway information, you can repair that damage and say, gosh, I saw 3 of 5 genes associated with this pathway. Maybe I should take a second look at those other 2 genes that were missing from my list. That's valuable. So robustness is something that you can gain from by using pathway and network information. And finally, sensitivity. If you are trying to ask, is this gene differential between these 2 cohorts, you may miss information that you could capture if you were looking across the data for all genes in a single pathway. So you're able to combine across more information and have fewer hypotheses to test by looking at things from a pathway perspective rather than a gene-by-gene perspective. Okay, so we can improve our sensitivity by making it possible to judge whether a biological pathway is differential rather than looking at it on a gene-by-gene basis. Where can I go for this? Well, the reality is that there's this huge spray of different databases out there. Every couple of years I need to sort of redo this slide, in fact, because I find that where these databases are hosted, what edition they're on, and so on, can change. So in this case, BioCarta and KEGG and Metastick and Pathway Commons, Reactome, STKE, Signal and Gateway, Wiki Pathways. I haven't validated these for this particular year, but in past years, those have been resources that were easily found and still live and kicking. These are going to have different perspectives on data. If you're interested in pathways, something like Pathway Commons or Wiki Pathways for that matter, is going to be more useful than something like reactome, which might be more useful in understanding small molecules, for example. But that said, most of these pathways and network sites play a lot of different games, and they give you lots of different looks at biology. So just because something is called reactome doesn't mean it also that it lacks all gene-gene interaction data. So the problem that's going to show up in several different cases here is one of limited coverage. So we can start with the idea that not every gene has a known function. This is true for the human genome, it's true for the E. coli genome. We've made efforts to try to get to complete information about individual genomes. Yeast is one that we really have tried hard on. And yet, we still find that for a given gene, we frequently have unknown function. You might ask, for example, do we have expression data for every gene called protein coding in the human genome? And the answer is no. We have some genes, even now, in the human genome that are putative. We think that they're expressed. Maybe we once saw a transcript from it, but are we able to quantify it? Not so much. So frequently we find that the gene or protein catalogs have incomplete annotation attached to them. And if only 50% of the genes for a given species are mapped to a function, well, some days you're just going to have to call yourself lucky that you do have that 50%. So these data are notoriously incomplete. Next, inconsistency among different databases. If you go to KEGG versus Pathway Commons, you're going to find that particular genes are going to have different annotations in those two databases. Previously, I've noted that resources like NCBI and Ensemble, and Uniprot for that matter, have come to an agreement about standard ways to name human genes. But how do we have the same kind of commonality in how we attribute functions to genes? Not so much. There are a lot of places where we find that functional information is quite inconsistent across these resources. There are, however, resources that have attempted, excuse me, I think it's called, I'm trying to think of the one that Ben pointed me to. I think it was called iPathway or something. This is one of the databases that's tried to reconcile what different resources had to say about genes for a given species and come up with common information that was supported by both resources. And finally, the relationship between pathways may be less well defined. That we have a really good idea about KEGG, but perhaps how it connects to fatty acid synthesis may be a little less clear for us. So finding these, as soon as you step out of these little boxes in which we put genes, you may find that the relationship to the next box is a little less clear. Okay, so when we talk about gene ontology, we start with the definition of ontology. Now you see, from philosophy, we get some very verbose descriptions. An ontology is a theory about the nature of being, or the kinds of things that have existence. So it's a little like a dictionary, in that it's a vocabulary that has definitions attached to it. But more broadly, it tries to talk about the relationships among these different terms as well. So biological ontologies generally feature controlled vocabularies. This is kind of the definitional part of it, and defined relationships. So gene ontology is designed around giving us the definition for the roles of genes and the gene products that come from them. In general, you'll find that gene ontology has 3 different hierarchies, all describing the same genes. So the first of these is molecular function, the second is biological process, and the third is cellular component. So if you were to look at dopamine receptor D2, you would see that its molecular function is listed as dopamine receptor activity. I think that's a pretty clear molecular function given the title of the annotation of the thing. Biological process is synaptic transmission. What's a synapse? Synapse? Do we have synapses? Neurons do what? Okay, good, yes. So we have neurons, and even after lunch, occasionally one fires a signal across a synapse, which is the gap between neurons, and that signal usually consists of neurotransmitters. We may all have to hit the caffeine. We will make it through today, though, I promise. So, we have a synapse, and synaptic transmission is a biological process that is mediated by the reception of a dopamine signal. Dopamine is a neurotransmitter. So you can see that these are both about what the thing does, but one is about a molecular function, one is more about the biological context in which it takes place. There's nothing in dopamine receptor D2 that says synapse, but by understanding its biological function is about transmission across synapses, we have more information about the context of its action. And finally, cellular component. Well, it's on a plasma membrane, you would kind of expect so, because synapses are these extracellular spaces whereby proteins in this cell membrane are communicating with receptors in this cell membrane. Okay, now I mentioned that we are going to have to hit some definitions along the way, and in this case, the types of relationships among different Go terms is defined by one of these four classes. It could be is a, it could be part of, it could be has part, or it could be regulates. If, for example, you know that one gene inhibits another, it will probably show up with a regulates relationship between them. Well, and the classes will. So we're going to look at a few examples from that. Now, I note that I've given a sort of incomplete list. There's actually is a, part of, regulates, positively regulates, negatively regulates, occurs in, capable of, and or part of. Well, we're not going to go right through the list, but I want you to think about how those play out. For example, I may have a intrinsic component of plasma membrane, and you see this P, that's signaling that that is part of a plasma membrane. The anchored component of a plasma membrane is part of the intrinsic components of plasma membranes. The anchored component of external side of plasma membrane is part of the intrinsic component of external side of plasma membrane. Some of these get a little verbose, right? But we also have is-a relationships. So an anchored component of external side of plasma membrane is a anchored component of plasma membrane. So you can think of the is-a as being sort of an equal sign between these groupings. So in this case, we're just looking at the is-a and part of. The regulates would be more like, as I mentioned, cases like gene inhibition. So you're going to also have a hierarchical structure to these different functions. So down at the bottom, you might see that you have programmed cell death. You have some collection of genes that are attributed to or associated with programmed cell death. But there are several kinds of cell death, of which programmed cell death is just one. So we see then that this is a subclass of cell death. There are plenty of single organism cellular processes. Cell death is one of them, so that hangs off of there. And single organism cellular process is both a single organism process and a cellular process, both of which are a type of biological process. So you can walk all your way from specific down at the very bottom of these hierarchies to general all the way at the top. That's true for all three of these, whether you're looking at cellular component, molecular function, or biological process. So as you get down, you get more specific. As you get up, you get to be like a professor where you give a very generic title to your talks, right? So grad students work down, no, that's a really unfair place to go. My point is that these hierarchies give us a structure in which we can understand a changing level of detail. As we move down, we get more specific. As we get up, we get more general. And all around, we see these is-a, regulates, part of relationships taking place. So how do we go about annotating using Go terms? Generally speaking, we have curators who are busily reading literature and trying to fill in gaps in our knowledge. Oh, for the first time we have someone who's associating this gene with a particular biological activity. So they get to read all this literature that we're turning out all the time and decide how to express it in terms of gene ontology. It may, however, be electronic annotation. Probably the most common example of this is annotation transfer. We learn of a new function within the human genome for a particular set of genes, And we then say, gosh, these gene sequences are similar to those for mouse. So we transfer the annotation from the human variant for the human ortholog to the mouse ortholog. So that's a very simple kind of electronic annotation. Excuse me. But these annotations are always attributed to a particular source, because we'd like to be able to track where did we get this bit of information from. They must indicate what evidence was found to support this Go term, whether it's a gene or a protein association. So there are lots of different experimental codes that we use. IDA, IMP, IgI, IPI, IEP. Computational codes, those all show up here. Author statements, whether you have a network curator, somebody who actually works for Gene Ontology, who's making this manual annotation, or you may have a statement from a researcher that says, I promise you this is associated with that. Here's a picture of the gel that proves it. And you have some other codes as well. So I'm not going to get into what all of these mean, but in each case they try to explain not just what this link is, but where it came from. All right, so what does that look like? Now, in this case, I've shown manual annotations and automatic annotations. This is one of the graphs that's available from Amigo. So, Amigo is probably how they want it to be pronounced, given that they're probably thinking about the Spanish term for friend, right? So, here we have human-made annotations in Homo sapiens, plus a whole bunch of automatic annotations that have been added. M. musculus, everyone knows what this is. Our favorite little mouse, right? What about Norvegicus? It's Norwegian, but it's not rabbit. No, that's not a rabbit. Rattus norvegicus. Yes, that's the rat. How about G. gallus? Anyone got that one? Why did the chicken cross the road? To get to the automatic annotations, clearly. All right. How about Damia rario? Anyone know this one? Rario, anyone? I believe this is angelfish. That's an angelfish. I'm not sure. It's been a while. All right, D. melanogaster. Somebody has this. The fruit fly. Right. Yes. When I was talking with my friends in the dorm when I was an undergraduate, and I was in genetics class at the time, trying to explain the difficulties of keeping these females separate from all of the males so that you could actually do a proper cross with them. And they started threatening to buy me a big hat with a feather in it. They called me the fruit fly pimp. It was really rude. Anyway, so yes, D. melanogaster. Right. And then, have you ever had the same feeling? I'm going to cross these. You're going to enjoy it. Anyway, so D. melanogaster, right. And then finally, C. elegans. Everyone's heard of C. elegans? Look at me, I developed. Great, okay, so that's C. elegans, right. So we have all of these different models. You can see some of them are very rich in annotations, mice, humans, rats. But with chickens, not so great, right? So we could do better on annotating how chickens genes interact. Zebrafish, I said angelfish. I meant zebrafish, I think. Zebrafish is Dana Ranferario, isn't it? She didn't check. One of you will know by the end of this lecture what Dana Ranferario is. Okay, and finally, C. elegans. So you might think that the more genes you've got, the more annotation you've got. But frankly, if you were to look at these plots for something like poplar trees, you would probably find the number of annotations is actually not that strong. We spend a lot of time in these model organisms trying to get a good roster of annotations. So we have interfaces within the Gene Ontology website itself, Amigo. We can use Bioconductor, our friend from our studio, and we can even use services like QuickGo over at the European Bioinformatics Institute. Lots of people care about Go. As a result, there are about a bazillion interfaces for working with it. If you decide you want to do Python interactions with Go, I'm sure somebody's got a library for that. So Go is just ubiquitous, it's everywhere. Now, I think this slide didn't update. That's really interesting. I think the one online is different. Well, I'm going to just go with it. I replaced this slide this morning, so maybe the PDF online looks different. Do you guys have a different slide there? You do? On your PDF? Ah, good. So she printed it. There's evidence I've been thinking about how to update this. So This slide has a simple point, and it should be easier to make. I really wanted to update this the numbers on this chart because I think this thing is at least 5 years out of date at this point. What we are looking at are the three sections of gene ontology: biological process, molecular function, cellular component. It's probably a good roster of things to know, isn't it? Should I write such an obvious quiz question? I guess I could, but anyway, which of these is not a legitimate fork of gene ontology? I could do something evil like that, but I lied too well, you know that. So here we've got Homo sapiens, here we have mus musculus, all right? And now we are looking for each of these three areas, what kind of annotation have we got? As I said, these numbers are out of date, but just think for a moment how this plays out. How many genes are there in humans? About 20,000. And here we see that for biological process, about 5,000 of them were missing information. We don't know what biological process they're part of. We saw that about 3.7 thousand of them were missing in molecular function. And we see that about 3.3 thousand of them are not, we don't know where they're found in the cell. Maybe it's just not expressed, right? So if Homo sapiens is missing this much information, how much worse could it be in a non-model organism? You see that the numbers are not all that different from mice. You might think, well, there are lots more ways that we can pester mice than we can pester humans, so maybe we know its molecular functions better. Not so much. Okay, but in the if you look at the slides that we have online, you will see that instead I've looked at the these three forks of Go for several species and asked, what is the origin of this annotation? Was it experimental evidence? Was it communication by the author, etc.? And it tries to show how this information builds out. And again, you will see that the number of annotations differs dramatically from organism to organism. Okay, on we go. So Go is fantastic. Everyone uses it. A lot of people, if you don't include Go, assessment of long lists of proteins or long lists of genes, will please request that you do include something like that just to make your results clearer. Sometimes that can be just as simple as the Go Slim analysis that we tried fervently this morning to produce, but the server was a little pokey. So a Go Slim analysis is just giving you kind of a broad approximation across major categories of biological function or molecular function, biological process or molecular function or cellular component, how these cells, how these genes separate for a long list. Now, KEGG is quite useful. I believe yesterday, was it no, 2 days ago, I showed you a slide in which caffeine was visualized. So caffeine sits in its own biosynthetic pathway, and we can see all these different processes that lead to it. Is there any case in which we use caffeine to produce another product? Yes, we see that we've got some lines leading away from here. We could use it to make theophylline, or we could use it to make 1,3,7-trimethyl uric acid, or we might even back up this process and create theobromine. So these pathways are a really valuable way for us to understand all the different ways in which metabolites end up interacting. And as we mentioned this morning, we have these very handy dotted quads, these sets of 4 numbers that are Enzyme Commission codes. These Enzyme Commission codes in turn help us to know what is the name of the enzyme that carries out this function. And because that information is carried over from species to species, one of the easiest ways to find orthologs between a pair of species is simply to ask if this gene has an enzyme commission code, what is the corresponding gene in this other species that has the same enzyme commission code? So that is a very straightforward way to say these genes play the same roles in biosynthetic pathways across these 2 organisms. All right. So molecular networks are usable in this context. I've included a link to KEGG up here, by the way, but it's always just pretty easy to do a quick search for KEGG networks. WebGestalt is very handy because it's really built around the notion that everybody needs a workbench. Maybe what you want to work with is Gene Ontology, or maybe what you want to work with is wiki pathways. There are all kinds of different networks out there, after all. So given that you did your work on a microarray, you've got these codes representing different probe sequences. You need to project them into gene sets. But where might you get those? So first off, there are lots of different possible origins for for these numbers. We've talked a lot about microarrays, and I don't need to say that this is the only kind of data that lead into this process. Maybe you're doing proteomics. If so, maybe you used UniProt to identify the tandem mass spectra to proteins. And now you've got some list of differential proteins. That is one of the kinds of translations that's embodied within WebGestalt. The software knows how to translate from a UniProt identifier back to a gene name for human, for example. So quite handy. RefSeq and Ensemble, these are other sources where you might get protein identifiers. Those two translate just fine. We have lots and lots of different gene identifiers out there. Maybe your numbers come from GenBank. You have a bunch of GI numbers, for example. Maybe you're working with Ensemble, and so Ensemble gene IDs are attached to these. All of these can be put on the same footing by the software, sometimes with variable results. As we noticed this morning, just because you give a gene identifier or a probe identifier to the software, it may yet fail mapping. It may not be able to translate that. Maybe there's a slight twitch in the software that put your probe names together. So it's always going to tell you about the extent to which mapping failed for your roster of differences. WebGestalt can then use that information to tie into Gene Ontology, different pathways. Here's pathway commons, wiki pathways, and KEGG. Different network modules, an approach we're going to talk about in a bit, such as transcription factors, microRNA targets, protein interaction modules, etc. There are lots of different ways that we can use these point-to-point interactions. And then at the end of the day, it's going to produce some pretty reasonable statistics. The statistical models within WebGestalt have all been published, they've all passed peer review. So if you use this method, you already have basically pre-written methods text to justify why this test was the right one. So let us imagine now that I have a set of genes that are known to be involved in development. This comprises my context. And I have another set of, I now have this other set of genes that are differentially expressed. So in this case, I may have a very large intersection here. I'm sorry. I talked about the list on the left as my context. This is not my reference. I misspoke here. These are a set of 842 genes associated with the development process. So this is not the full set of all genes that were detected. So now I have all genes known to be associated with development, and I have the set of genes that are ones that I've found to be differential. I count the number of genes that appear within this pathway, or within this biological process, that are also part of my difference list, and I get the number 98. So is 98 a good number, or is that a bad number? This is a difficult question to answer. So we can ask, if we were to choose 581 genes at random across the entire genome, what number would we have expected by random chance to come to match with this set of development genes? That number is 65. So we have about 50% more genes attributed to this pathway than we would have expected by random chance. Putting together a p-value for that is a little different though. So the structure we use for this is called a contingency table. I talked about them before with Fisher exact test, and here it's showing up again in Fisher exact test. So the Fisher exact test is about producing a cited p-value. What is the number of genes that we saw in this case that overlapped with the development set? I think it was 98, right? So we want to compute a probability for getting exactly 98 hits in this pathway, exactly 99 hits, 100 hits, 101 hits. So basically we're looking at more extreme results than what we saw. So that, if we add together all of those probabilities, we get a p-value that says what's the probability that we would have seen this many or more hits by random chance alone. If you're only computing the probability of this particular outcome, you can use the hypergeometric distribution without adding up all those probabilities. And the formulation for it looks something like this, the k values there, the n's, the j's, etc. So the contingency table ends up looking a little like that. That you can ask how many genes are in this subset that were observed, how many genes were outside this subset that were observed. In general, at large, how many genes are found in this pathway, and how many genes are attributed with something other than this pathway. That set of 4 numbers gives us a probability or a p-value, depending on whether you're doing this through the hypergeometric distribution, just one-time application, or the Fisher exact test if you're computing the cited p-value. But contingency tables allow us to compute these probabilities in the hypergeometric distribution. Okay, hypergeometric distribution is quite a mouthful. I mean, people use it all the time for all kinds of questions. Frankly, whenever I talk about hypergeometric distribution, I think about the fact that I have this jar of marbles beside my desk of black and white marbles. So we talked about it before when we were asking, for example, how might you score a match to a sequence for a tandem mass spectrum. So this is just one of those statistical models that shows up everywhere. Okay, now I mentioned that we have complex hierarchies coming out of gene ontology. This is an example of that. So in this case, we see that response to unfolded proteins, this is a biological process right here, has 12 genes in it. In the case of this particular overrepresentation analysis, we see that these 12 genes are giving us an adjusted p-value of 1.32 times 10 to the negative 8th. That is a vanishingly small probability. And in fact, these have been adjusted for multiple testing already. So we feel very strongly that response to unfolded proteins is a real hit for us. But I want to note that these genes are also part of this set of biological process, which means that the same genes that are causing a low p-value for this category are also contributing to a low p-value for that category. This is the correlation I'm talking about between different p-values that get computed. So when we see something like this at the bottom, inspiring this to become a low p-value, and those above it to become low p-values, and those above it to become a low p-value, we have to remember that these are not independent hits, because the same genes that make the most detailed feature significant are tending to make the features above it also go significant. All right, you can do the same sort of thing within pathway analyses like this. So in this case, I might have a set of input genes that I've claimed are differentially expressed. And in the case of KEGG, I can see that all of these genes are found within this TGF-beta signaling pathway. Now I want to note that not every gene in that pathway has been declared differential by us. Just a subset of these, just these blue ones. But just the same, that number of hits can still produce a significant p-value, that we see that more of these genes are included in our differential list than we would have expected by random chance alone. So you can do this against Go, you can certainly do this against KEGG just the same. All right. Now it is possible to do this in the network module space as well, but I want to point out that the annotation here is it can be a fair bit messier. So we're still talking about overrepresentation analysis. But here we see that we have one of our lowest p-values is right here. This is 3.58 times 10 to the negative second. So that's something like 0.035 after adjustment. Okay, in this case, these genes are also part of this next higher module. But you see, before we had these nice clear-cut relationships. For example, in Go, we saw that this was response to unfolded protein. Underneath that, it's response to protein stimulus, etc. We can walk our way up the tree and see that we're getting more and more general. In the case of network analysis, though, we frequently run into issues like this where we see that these differential genes hitting this module are in turn part of a larger scale module, which is part of another larger scale module. The relationships aren't quite as clear. We can ask, how many genes overlap between these, etc. But these don't come with very handy explanations as to why they're more generic, larger scale models. One of the reasons for this is due to the nature of biological networks. We're going to talk about it in a few slides, but I just want to point out that you can use overrepresentation analysis with biological networks, not just with pathway databases and gene sets like gene expression. Like Gene Ontology. Sorry. So overrepresentation analysis gets used an awful lot, but there are some limitations to it. So we start with this idea of thresholding. We have seen that we've seen that the p-values from these can look very tempting, right? A nice 0.03 having survived multiple testing correction gives us something we can write home about in our manuscripts, and that's always lovely. But where do you set the threshold? Do you need to set a different threshold for claiming the second box within the hierarchy, or do you need, or should you only allow for one box in which these genes occur to be called differential? Some systems like David over at the NIH website, which can also do this over-representation analysis, have been complained about by people who say that it tends to call not only the bottom node significant, but the one it borrows from significant, and the one above that significant. That's just kind of crazy. We need to recognize the relationships that the same genes appear at multiple levels of the hierarchy and thus can drive correlated p-values. So where you set your thresholds, how you pick which ones you're going to talk about in the manuscript can end up being a little messy. Next, one of the things that we worry about is that when we produce our difference lists, we tend to apply some threshold. And if something is below that p-value threshold, well, we say it's differential. And if it's above that p-value threshold, well, we say it couldn't possibly be differential. But the reality is a lot fuzzier than that. We have things that are, imagine that you have something that has a p-value of 0.04 after adjustment, and another one that has a p-value of 0.06 after adjustment. Are you ready to say that the one that's 0.04 is absolutely, certainly significantly different, and the thing at 0.06 is absolutely not possibly different? The p-values are not that far apart. In reality, our claims about which things are differential and not are putting a hard dichotomy on things that we don't really have enough information to say are definitely or definitely are not significantly different. So imagine that you could instead use the p-values to rank genes, to help you say, well, something that's 0.04 I'm going to trust before I trust something with a p-value of 0.06. But we're not going to apply some hard threshold to it. So now we're prioritizing things with high p-value, with very low p-values rather. But we're not going to say that there's some hard and fast rule after which they're certainly not differential. So this is kind of a way to use the ordering. We would like to have a way to use the ordering of genes on our significance list and kind of throw away this is or is not significant ruling. We'll get to that in a minute. Next, treating pathway genes as a set frequently ignores the fact that some of these genes are quite central to how this pipeline functions. When we looked at these plots in KEGG and in Go, we don't really see a lot of pathways that have product A, then product B, then product C, then product D, then product F, and that's the only pathway through there. We frequently see that there are multiple arcing ways to get between metabolites in these KEGG plots. So if you are to put all of those in a little box and say, these genes are all associated with fatty acid metabolism, you're kind of ignoring the fact that some of them are tenuously associated with fatty acid metabolism, and others are quite central to fatty acid metabolism. So using these kind of concrete boxes is a bit of a problem on that score. So let's solve one of these issues and talk about Gene Set Enrichment Analysis. The idea here is that we're going to have a sorted list of genes, those that are most differential to those that are least differential, and use this to find which pathways are non-significantly linked to this rank-ordered set of genes. This was created by Jill Messeroff and colleagues over at Broad Institute. So gene set enrichment analysis is effectively giving our gene set and asking which ones are grouped together in these orderings. So imagine this as, this end is most up in a cohort versus cohort study. And these down here are most down in our study. So if we see if we're thinking of a particular pathway, we can ask, where are the genes associated with this pathway found on this list? If they're on the most up end, maybe we have stripe, 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 and they're all packaged together. That looks really good for us, because the most differential genes are strongly associated with this particular pathway. If, however, they're spread all through here, or you find them at both ends, that's kind of odd, isn't it? Because once you have something like that, the evidence is equivocal. You have some genes associated with this process way up, and others that are way down. So you can imagine then that you would like to get a whole cassette of these genes associated with a particular pathway grouped together all on the up end or all on the down end. And from that, you can then ask basically how the evidence compares to this enrichment score. So if you find that all of the genes that associate with this particular pathway are all grouped together at the up end, you're going to get a really good deviation upward on the score and end up with a strong enrichment score for that pathway. So it's a bit non-parametric in that it's not going to require that genes be significant or insignificant. It's going to treat them as being on this very up to very down scale and look for the genes associated with the pathway to be clustered together. Okay, so generally speaking, we've been talking about these sort of canonical databases that have a whole bunch of genes on a list saying these are attributed to this biological process, molecular function, or cellular component, and that's great. You can use all sorts of pathway databases for that purposes. You can use Gene Ontology for that basis. Over representation analysis is a very standard way to do this. WebGestalt is a very easy workstation on which to compute these. Gene Set Enrichment Analysis, is a little more computationally demanding, but it has proven to be a much more robust way to evaluate data from, say, microarray experiments than going after the genes that are differential. So when we do this, our existing knowledge is what's at play, and ultimately we don't have as much knowledge as we'd like about the functions taken on by each gene in the genome. So that's going to be a bit of a limitation. From there, we step into this fine world of biological networks. Suddenly, instead of having a set menu, this pathway has these genes attributed to it, we're going to be working in sort of an inferred network arrangement instead. So when we talk about a graph, we're talking about nodes and edges. This morning we looked at GraphViz as a way to build our custom graphs from a language designed for describing them. In this case, though, we're looking at them more generically. We have a vertex, which is one of these biological objects sitting in space. Maybe it's a gene, maybe it's a transcript, maybe it's a protein. We then have edges that refer to some of this annotation we have about it. This gene and another gene are known to correlate, therefore we put an edge between them with some sort of label indicating this. The evidence here comes from microarray correlations. Sometimes, though, they're directional. If you have two genes that correlate, you don't say that A correlates with B without also saying B correlates with A. It's a symmetric relationship. But sometimes you find that the relationships between biological compounds are directional in nature. Consider that of a transcription factor. We say that this transcription factor has an impact on the transcription of these genes, but we don't generally find that the same relationship operates in reverse, except for things like inhibition. So in this case, we have a directionality associated between this gene's product alters the relationship, alters the expression of these genes. That's a directional relationship. A concept that shows up a lot in graph theory is degree. When we have many edges, you may find that you have a singleton, which is to say a vertex that's sitting out there all by itself with no edges. You may have a pair of vertices, of nodes, that have an edge between them. So you would say that the degree of each of those is 1, because it has just one edge connecting it. You might find something like a triangle existing in space. A triangle is technically known as a 3-click. We have 3 nodes, and they have every possible interconnection among themselves. Each one connects to the 2 others. We could step up to a 4-click, where you've got 4 genes, and each of them has a complete set of relationships to all the others. So these are all examples of connected components that we're seeing at the bottom. But when we talk about degree, we're asking how many edges lead into or out of this point. So in a triangle, in three genes that are all completely connected, we would say the degree of each is 2, because each connects to 2 other items. This degree concept is something that shows up pretty heavily in advance. So I mentioned clicks a moment ago. There's a formal definition here. So I'm going to just borrow our chalkboard for just a moment. If I have a gene A, and I have a gene B, and I have a gene C, and I have this, I do not have a click. In this case, A connects to both B and C, but we don't call it a click because B lacks a connection with C. A lot of us are more familiar with the term click from the playground, where the cool kids would play with each other, but they weren't going to play with me. Definitely not, right? So the click on the playground is sometimes a very useful model for thinking about how genes interact. So in this case, we don't have a click. Once I add this relationship between B and C, now I have a 3-click. If I have a 4th gene, have I made it to a click yet? I haven't, because I'm missing one interrelationship. A and D must be connected for this to be called a click. And what do we call a 5-click? A pentagram. That's right. Yes. So if you're doing any devil worship, then you can add your E in here, throw that in there, push this here. Now you got a pentagram, right? So the 5-click is the pentagram. All right. Now that we've finished all that awfulness. So we use this click terminology as a way to represent tightly connected. That is the most tightly connected set of nodes that you can have in a graph, a click. And the larger the number, the less likely it is to happen by random chance alone. Okay, modules. So if you have a set of genes that are much more tightly connected to each other than they are to everything else around them, we call that a module. Path length is one of these terms associated with a term that we saw early in this talk, the random walk. Does everyone remember that? So I'm going to move back over the chalkboard. Imagine we've got a 3-click over here. I've got another few items out here. Maybe B is connected to E, C is connected with D, and over here we have F. We would like to make an assessment of how much A and F relate in function. So one of the ways we might do that is to ask the path length from one to the other. So what is the minimum number of edges you have to traverse to get from A to F? 1, 2, 3. Is that the only path? It's not. You could also go A to B, B to C, C to D, D to F. That's a longer path. So when we're asking about the relationship and function between different genes, a shorter path implies a greater relationship. Okay, so path length is one of the ways we go about this. The concept that shows up all over the place in biological networks is that of a hub. So when we have a particular node, particular gene that connects to all kinds of other things, that's a gene that plays a disproportionately important effect in that biological network. We'll talk about some examples, but things like, say, P53 tends to interact. It has lots of interaction partners. As a result, it plays kind of a hub-like role. Okay, so we have lots and lots of possible networks that we might derive here. We may have protein-protein interaction networks. We may have signaling networks, gene regulatory networks, metabolic networks, co-expression networks, and genetic networks, genetic interactions, gene-gene interactions. So with each of them, it's valuable to think about what kinds of biological principles these nodes represent, whether they're genes or proteins or transcripts, or metabolites for that matter. We want to think about, are the relationships symmetric, such as a gene microarray correlation property? Or are they directed, meaning that one affects the other, but not necessarily the inverse? So I really think this slide is a really, really valuable one to know. If you're going to know anything about biological networks, I actually would really recommend you take a read of this paper from 2004, because it, I think, causes us to rethink a lot about how biology functions. So I'm going to try to walk through this kind of carefully. We start with scale-free. Scale-free is a discussion about the to talk about the scale-free property of biological networks is to talk about the frequency of degrees across the nodes in biological networks. So what was the sorry, I just lost my term here. What is degree? What do I mean by degree for a biological network? The degree is what? Number of edges for a given node. So in the graph we drew earlier, B is pretty well connected. It connects to three different properties, or three different nodes. C is pretty well connected. It also connects to three different properties. We could make a graph of the distribution across this little network I've drawn. E has a degree of 1, F has a degree of 1, D has a degree of 2. Put all of these together and you can say what is the distribution of degree for that graph. So when we look at scale-free networks, this is the property of biological networks, we find that some genes are just ludicrously connected and many genes are only minutely connected. As a result, those genes that have lots and lots of edges leading in and out of them tend to be massive points of failure for the network if that gene is mutated in a way that alters its structure. All sorts of biological processes are disrupted. So you might think about what are the genes that you can knock out that an organism will still grow with versus those genes that you knock them out and you're not going to get any cells surviving. Right? That's probably a bit of a proxy then for which of these genes have lots of interactions versus which genes have very few. A gene with one interaction is a whole lot more likely to still produce a viable organism if you knock it out, as opposed to something that's got lots of interactions. So that's because biological networks are scale-free. They have a few nodes that have huge numbers of interactions. They are also small world. Has anyone ever played the game 6 Degrees of Kevin Bacon? Kevin Bacon? Anyone know the actor Kevin Bacon? I realize I'm old, but he's still acting. So, all right, you might remember him from Footloose, or you might remember him from X-Men First Class. The man's been in a lot of movies. So, one of the games you can play in Hollywood is to ask what someone's Kevin Bacon number is. If they have performed in a film with Kevin Bacon, their Kevin Bacon number is 1. If they performed in a film with somebody else who performed in a film with Kevin Bacon, their degree is 2. If they performed in a film with Fred, and Fred performed in a movie with Ginger, and Ginger performed in a movie with Kevin Bacon, their number is 3. So you can see that there's this degree of separation is set up in this way. So what we find is that if you have a reasonably connected gene, I mean a singleton, this isn't going to work, but something that's connected to something, we frequently find that we can step from its cousin to its cousin to its cousin to its cousin and get to some major protein. So we can ask for 2 arbitrary genes in a genome. What is the number of steps we need to take to get from one to the other? And frequently there's a relatively small number of steps. That's partly because of this hub-like relationship. And finally, it's hierarchical and modular. So we've already seen these hierarchies in the form of Go terms. Remember that the most specific Go terms down at the bottom are part of Go terms that encapsulate that and other activities at a higher level. This reflects the fact that our biology has a modular structure. We have lots of interrelated genes that function all together to create a process. But at the next higher level, we find that this process is one of several processes contributing to a larger process. And that in turn, that larger process ties up with other larger processes to do even bigger functions. So when we think about biological networks, we should think about the fact that a few genes or a few transcripts or a few proteins have many, many interrelationships, that one can find one's way from one pathway to another by kind of stepping from one partner to another, and that boiled together, biological processes are hierarchical in nature. Now, there are lots of great tools out there to do network visualization. I used to have a big table on this slide that kind of irritated me because it just didn't have hyperlinks and stuff like that. So as an exercise, I started working my way through the 20-row table to figure out which tools were still live. And a lot of them had gone out of use. Biological network tools come and go pretty quickly. And yet, we find that tools like GraphViz, Vizant, Medusa, Ondex, Pagic, BioLayout Express, they're still live, still kicking. But I want to point out that one of these tools has a huge footprint in this space. Because it's 3:30, I'm not going to tell the story at great length, but I just want to say that when I went to grad school, my department, Molecular Biotechnology at the University of Washington, was recruiting graduate students for the first time from the outside world. Before, they had always pulled students from bioengineering and said, okay, you're now a molecular biotechnology student. But this year, they were going to the outside world to bring students in. And they recruited 2 students. They brought me fresh out of an undergrad. I just graduated from the University of Washington with a degree in biology. And they recruited another student from MIT who had just completed his master's degree in electrical engineering. Which of those students had a clearer notion of what he wanted to do with his life? The master's student. So for years, my classmate in graduate school was Trey Eidegger, a very, very bright guy who later went on to create Cytoscape, the premier tool for visualization of biological networks. So if you think you have it bad in going through grad school with very talented people, I just got to say I got you beat. Trey Eidegger is a full professor over at UCSD. He is ridiculously busy, goes all over the world, gets all kinds of grants. He's an amazing scientist. So Cytoscape was his brainchild. And I really can't do it enough justice here, but let me just say that it allows us to integrate data from biological networks and certainly to visualize them very, very effectively. There's any number of papers that you could read about this topic. Cytoscape's been around for quite a long time, and it seems to be getting more and more powerful every year. So if you want to visualize very complex interrelationships among different pathways and networks, I strongly suggest you give it a look. It can probably produce you a great visualization for your next poster, or even better, for your next manuscript. Okay, now I've already alluded to this, but I wanted to have a slide devoted to this topic. Sometimes we want to know about the relationship between a pair of genes. In general, if we look at gene-to-gene interactions, we find that the further things are apart, the less related they are in function. And conversely, if there are fewer hops between them, we find that they have a greater and greater functional similarity as well. If you require 8 steps to get from one gene to another, the Go terms that explain these 2 genes are going to have very little intersection with each other. So frequently we find that this the greater the proximity of relationship, the greater the functional similarity. One of the things that becomes possible to do is to build linear relationships of genes so that the network modules are listed together by linearizing these graphic maps of things. So in this case, we're looking at some software called NetSam that is part of Bing Zhang's visualization toolkit. Bing Zhang's the guy who brought us WebGestalt. Well, he used this relationship data to give us something called NetGestalt that's not dependent on these standard categories of proteins like Gene Ontology. Instead, it's capable of working with gene-to-gene and protein-to-protein interactions. This is the software, by the way, that I was trying to get a demo working on today. I found that the software was hanging for me, so I'm sure it'll be back up. But you can see then that the software has created these sets of tightly related genes. Imagine, for example, ABC from our chalkboard exercise earlier. You can zoom in on them, for example, to look at, in this case, we're looking at this very small region, so this network module in effect. We zoom in and see that these genes fall within that category. We're able to look at statistics that have been overlaid on these, reflecting your data. So if you have microarray data, you can project them using NetGestalt. And it helps you to organize all of your data very well. What if you have, for example, proteomics data and microarray data, and you want to say, are the p-values lining up for genes, or are they completely distinct for the two? This software toolkit will let you display both on one common reference frame, all aligned by gene name. So it's quite powerful, and we use this within the clinical proteomic tumor assessment for National Cancer Institute Center, National Cancer Institute. So CPTAC was this group I was part of for 10 years. NetGestalt ended up being our premier workbench for displaying our results because we were showing genomic data aligned with proteomic data. So building your assessment on pathways and networks will help you in biological interpretability. At the end of the day, most biologists don't want a laundry list. They want some sort of biological enrichment. What is the functional implication of what you are showing me? So doing this through Go or doing this through KEGG is one of the most basic things you can do to improve interpretability of your data. There are also statistical means that allow us to more readily discover these biological differences by doing a p-value per biological pathway, for example, rather than per gene. We tend to think of biological pathways as being built from sort of a categorical analysis. This collection of genes is referred to as the set, for example. Biological networks are a more graphical way to think about this, often built on point-to-point interactions from these data. Gene Set Enrichment Analysis, overrepresentation analysis are just two of the most common statistical tests that we use for showing enrichment of biological function in our data sets. It's worth your while knowing the strengths and weaknesses of each of them. So with that, I have given my 8th lecture for you in bioinformatics. It has been a great pleasure. Thank you very much for this time with you. My door is often open. I'm usually here Mondays, Wednesdays, and Fridays. I visit other campuses on Tuesdays and Thursdays. But I'm always happy to set up an appointment with you. If you need help figuring out what to do with this data set, by all means, write me a message. Maybe I'm not the right person to answer that question, but I will try to make sure you get connected with a person who can. Thank you, everybody.
Thank you.